Hello everyone, respected teacher and dear student. Welcome to student presentation week 2025 on Pharmacognostic and Phytochemistry 2. I am YASHASVI JANRAO Library student of B Pharma 5th Sem from PR Patil Institute of Pharmacy Talegaon. Today I am giving a presentation on glycosidic content plant that is SENNA. This is the photo of SENNA plant with yellow flower and green leaflet. So that is start with a brief. What we looking through this presentation of Senna. Firstly, we start with the synonym of Senna, then its biological source, its geographical source, collection and cultivation, microscopy, macroscopy, chemical constituent, extraction and isolation, its analysis, its uses, adulterant, marketed preparation, and lastly the acknowledgement. Now, let's start the presentation. The synonyms which are widely used for the Senna are: first is Alexandrian Senna, second is Tinne Senna, and third is Folia Senna. Next part is biological source, from where to which part of the plant to be extracted. Senna leaf consists of dried leaflet of Cassia acutifolia, known as Alexandrian senna, and of Cassia angustifolia, which is commercially known as Tinnevelli senna, and which is belonging to the family Leguminosae. Further about the geographical source of senna leaves, where it is found. Alexandrian senna is indigenous to South Africa, Egypt, and in the middle of the upper territories of the Nile River. Indian and Tinnevelli senna is indigenous to South India and cultivated largely in Tinnevelli and Ramanathapuram district of the Tamil Nadu. Next, how the plant is cultivated and collected. Senna leaves is 0.5 to 2 meter of height, which is mostly cultivated in level and clay semi-irrigated lands. Lifespan of cultivated plant is 2 to 3 years, but it is grown annually. Alexandrian senna sometimes mostly from the wild, but sometimes can be cultivated. The cultivated crop become ready within 2 months, but the leaves are plucked in 3 months. Let's move forward to its macroscopy. While studying the microscopy of senna leaves, it includes the study of its color, odor, taste, size, and shape of senna leaves. The color of senna leaves. The color of senna leaves is grayish yellow for Alexandria senna and yellowish green for Tinnevelli senna. Now, the odor of the senna leaves. The odor of the senna leaves is light and characteristic. The taste. The taste is mucilaginous bitter due to the presence of mucilaginous. Size. The size is 25 to 30 mm in length and 7 to 8 mm in width. The lamina is found on the both surfaces or the both sides. Now the shape. The shape of the leaves are lanceolate. The entire apex is acute with spine at the top of the bases of the leaflet are asymmetrical with the transverse line. The more prominent on the lower surfaces while the trichomes are present on the both sides of the leaves. Next is microscopy of senna leaves. Senna represent the typical histological character of isobilateral leaf. The epidermis shows presence of unicellular, conical, thick-walled worted trichome. The trichomes are slightly curved at their bases and are present on the both surfaces. Parasitic stromata are present on the epidermal surface. The palisade tissue is present on the both sides, consisting the rectangular cell, including the clustered cluster of calcium oxalate. Next is the chemical constituent of senna leaves. Firstly, we see the chemical constituent in the leaf. It contains anthraquinoglycoside in the lipsinocyde A and B based on the aglycosinidin A and sinidin B. Sinucide C and sinucide D, which are a glycoside of heterodianthron of oleimidin and rin. Others include palmidin A, rinanthron, and oleimidin glycosides, some pre-anthraquinones, and some potent compound of acid undetermined structure. Cassia usually contain greater amount of sinusides, naphthalene glycoside, flavonoids, mucilage, pentyl, and reducing sugars. Now we discuss about the chemical constituent in the fruit. It contains sinucide A and B, naphthalene or glycoside like tinnevelline glycoside, and 6-hydroxymucin glycoside like mucilage, flavonoids, volatiles, sugar, and resins. Prefer the picture given on the slide for the better understanding. Now, about its extraction and isolation of senna leaves. For the commercial purposes, calcium cyanide in different states are extracted for the purpose of stability of compound. The drug powder either treated with the 90% of methanol or 80% of acetone for 6 hours and then for 3 hours with cold water, when leads to an extract containing about 70 to 80% cyanide. This method is enabled to extract about 625 cyanide by the use of solution of non-ionic surfactant and polyethylene glycol in 70% of ethanol. Cyanide and other anthracene derivatives can be extracted. Now about the isolation of cyanide. For the isolation of cyanide, the use of non-polar synthetic resin with spore structure. In another method, senna powder is treated with citric acid in methanol, followed by extraction with methanol toluene mixture of ammonia, and then subjected to treat with calcium chloride. Let's move forward toward the chemical test of the senna leaves. For the chemical test of the senna, Born-Traeger test is applied. Drug is boiled with the dilute sulfuric acid and then filter it. The above filtrate is treated with either benzene or ether or chloroform and shaken well. The organic layer is separated in the test tube and ammonia is added through all of the test tube. Pink to red color of the layer shows the presence of the anthraquinone. For the better understanding, prefer the picture given on the slide. Next is uses of senna leaves. Senna leaves are used as laxative. Senna leaves is also useful in loss of appetite, hepatomegaly, splenomegaly, indigestion, malaria, jaundice, and anemia. It is also useful for irritable bowel syndrome, in short form IBS, and loss of weight. In cosmetic field, senna improves the skin quality by moisturizing and softening the skin. It can be also used for protecting and repairing the damaged sensitive skin. Let's move forward toward the adulterant of senna leaves. The adulterant of senna leaves are following. First is dog senna, second is Palte senna, and third is Arabian senna. It is all type of adulterant are adulterated with Alexandrian senna as they contain very low amount of cyanide in them. Further about the marketed preparation of senna leaves. There are many marketed products of senna leaves. Some of the following. Marketed product as Syned, Prusinid and Ixlax. Syned tablet is sold under brand Cipla. Exclax is sold under brand GSK, and lastly the Prusenate is sold under the brand Novartis. And lastly, about the acknowledgement. I express my heartfelt gratitude to Dr. Nilesh Banarase sir, our esteemed guide, for his continuous support, motivation, and valuable guidance. I am thankful to respected principal sir, director sir, all the faculty members, and non-teaching staff for their encouragement and assistance. Special thanks to my friend and classmates for their cooperation and moral support. Finally, I just want to say that senna leaf is a nature's powerful remedy for digestive health. Thank you everyone for watching my video.